Okay, I think we are good to make a start. So, welcome everyone in the room, and welcome to everybody online as well. Thanks for joining for this session. So the idea behind this session essentially is, as many of you in this room will be aware, there's been a growing interest in causal inference generally, and a growing community of people interested in causal inference, having expertise in various aspects of causal inference, in both from a sort of statistical perspective and applied perspective, and more of an AI machine learning perspective. At the same time, lots of initiatives going on that I'll talk about shortly, and so we thought it was a really good opportunity to bring together the community in Manchester and beyond. We have people from elsewhere, certainly across the north, so it'd be really nice to hear what's going on. So I should introduce myself, of course. So I'm Matthew Sperrin. I'm a senior lecturer in statistics, so I have a background in statistics. I'll say a little bit shortly about how I got interested in causal inference. But just to sort of take us through the day, so this is just a brief welcome session. We've then got a series of short talks from across the university, across the group. We'll then have a coffee break. There'll then be some very short talks from some early career researchers, PhD students, etc. We've then reserved around half an hour at the end to just talk in the tables about challenges in causal inference. So we've got some prompt questions for that. And then we'll just finish with some closing remarks, and then you'll be pleased to know, and I guess that's why many people are here, is there's some free lunch available at the end. Okay, so I'm assuming, I haven't heard otherwise, so I'm assuming the sound quality is good online. This room, so fortunately for the people over there, the way this room works, it's a little bit of a funny shape, and the speakers have to stand all the way over here. But I hope that the acoustics are fine for people at the other end, and you can still hear okay and just about make out the people that are talking in the distance. There's still there's seats just down at the front here. Yes. So just to set the scene, I suspect that we've got a mixture here today of existing experts in causal inference, along with people who are just interested in causal inference, along with people who are just here for the free lunch. So I'll start off by just saying a little bit about causal inference, just at a very basic, non-technical level. So a paper that I often referred to is this one by Miguel Hernan and colleagues. So Miguel Hernan being one of the giants of causal inference, especially in the medical space in Harvard. So he wrote this paper, which is more of an education paper, really. So a second chance to get causal inference right, a classification of data science tasks. So data science and AI are sort of, the big words that we use. But essentially, whenever we're doing data science, statistics, AI, machine learning, whatever terms we want to use and whatever we mean by these terms, then really we're usually doing one of, or perhaps some combination of, three tasks. So the first one being description, where we're just summarizing data in some way to provide some useful information. So taking a large collection of data and summarizing that down. The second task being prediction, and this is probably the mainstay of, is often what people think about when they're talking about machine learning algorithms. So often you're talking about a supervised problem where you have some inputs and you have some output that you want to predict. So predicting future risk, for example, in a healthcare context, or predicting future stock prices, or anything where we're predicting something about the future. So the point about prediction, and I'll just say this in a slightly naive way for now, we can get into nuance later, but in some sense with prediction, we're not really interested in what these inputs are. Well, we're potentially interested in what they are, but We don't really care if there are things that really cause the outputs. We just have a collection of inputs that we're sort of putting into an algorithm, which might be a black box algorithm, and pops in an output. I should say that that's not what I really think and not what most people doing prediction really think, but I'm just sort of emphasizing a dichotomy here. So causal inference, or in this paper it's called counterfactual prediction, but I'll use the term causal inference here, is where we're using data to predict features of the world as if the world had been different. So here we're asking a question about the way things could have been if something had been different. So it's often a hypothetical, it's often a counterfactual question. And we're often interested in contrasts, right? So if I, the classic example, I had a headache, I took an aspirin this morning. If I hadn't taken the aspirin, would my headache have got better? Would it have got better quicker with the aspirin, etc.? And those kind of questions need us to understand much more about the structure and the mechanisms behind what's going on. We can't just throw in inputs and get out outputs. Life is a bit more complicated when we want to do causal inference. So yeah, so causal inference is, and again, this is probably a bit of a provocative statement, but causal inference is harder in some sense than description or prediction. Harder in the sense of having a deeper understanding of the data generating process. So we can't just throw ever fancier and fancier deep learning algorithms at a problem. We need to have some understanding of the problem we're trying to solve. We need expert input to learn about what the causal pathways might be. The usual sort of, historically, the sort of standard solution for doing causal inference is randomized controlled trials. So that's, this is why we do randomized controlled trials, because by randomization, we eliminate confounding, and as long as the trial has been done well and there aren't any issues, then we can essentially interpret the output of that trial as a causal effect at an average level of that treatment. But randomized controlled trials obviously have their limits, and that's why increasingly we're interested in the potential of using observational data to do causal inference. So there's an interesting history with using observational data to do causal inference. For a long time, it was kind of considered taboo. And if you ever mentioned anything to do with causality with observational data, that was just considered not appropriate. So sort of in the history of statistics for a long time, that was, we could only find association. We can't find causation. But more recently, there has been a bit of a shift to saying that, well, if you're looking for causal effects, you might as well be upfront and say that, and be clear about what assumptions are required to estimate those causal effects. Probably one of the most important, well-known assumptions is that you know what all the confounding effects are and you can adjust for those. But we might as well be upfront about that. We shouldn't shy away from the fact that causal inference is really what we want to do. So increasing interest in when can we do causal inference with observational data. How important are the assumptions that we need to make? How realistic are they? Usually not very realistic. What other ways can we do this? How can we triangulate across different sources of evidence, like Mendelian randomization and things like this are very popular to give a different window. Combining with trials, trying to emulate trials. There's tons of activity in this space on how to solve these kind of problems. So just as a quick example of stuff that we're working on, so this is Alex. So Alex at the back in the bright orange jumper is working on this project called the Chariot Project, where we're essentially interested in cardiovascular disease prevention. The usual tool that's used in the UK in primary care is Q-risk, which estimates risk of heart attack or stroke over the next 10 years. But that's, quite deliberately a non-causal model. It's a model that, it's a prediction model. It just tells us what that future risk would be. Unfortunately, and I'm sure Brian won't mind me saying this, that Brian and his colleagues, so Brian's a GP, thought for a long time that it was fine just to sort of plug in, what if I stop smoking? Okay, well, we'll change the input to ex-smoker from current smoker, contrast those two predicted risks, and that's telling us something about risk of stopping smoking. And of course, that doesn't work. I mean, it doesn't take too long to sort of think through the problems with that. So the unmeasured confounding, the mediation effects that you're not correctly capturing, etc. So these models just aren't designed to estimate those sorts of causal contrasts. So in the project that Alex is working on, Chariot, we're trying to address this and build a model that will allow some of these causal contrast questions to be answered. So that's perhaps for the applied people, a very practical kind of question that we're interested in answering using causal inference. And you'll hear about many more throughout today. So as everyone's aware, there's a huge explosion at the moment of interest in AI in particular, whatever we mean by AI, but typically I think what people mean when they talk about AI is either deep learning or generative AI in the context of things like ChatGPT. And importantly, the AI is by default non-causal, so we're not building causality into this. And that causes all kinds of problems. So one of the most sort of well-known problems corresponds to this little picture of the chest X-ray here. So there's some sort of token here that is used in the image to actually a sort of post-classification inadvertently of a particular diagnosis. And the AI obviously learns that this is here and uses it as a shortcut to build a diagnostic model. So if you actually look at what the AI model is doing, it just looks for this token. If the token's there, then you have the disease. If it's not there, then you don't have the disease. So lots of stuff like this going on. And thanks to Sotos for this slide. And that leads me into CHAI, which Sotos, who's responsible for the previous slide, is the PI on Edinburgh. We have a spoke here in Manchester, and CHAI is the Causality in Healthcare AI hub, where the interest is in how can we build causality into AI. So, doing AI but on a firm statistical and causal foundation, essentially. So that is. They sent me some swag, so I've put the swag out. So help yourself if you want a mug. We're very interested in this in healthcare. So there's a series of research challenges that we're looking at, notably, how do we predict under intervention? How do we do causal discovery, etc. And we'll come back to this at the end, but there is, perhaps this is a sort of follow-up potential, is that there is an events and workshop fund that's just been launched that is obviously quite applicable to this space. I'll show this slide again later. The other thing I just want to cover as well is, so we're here in the Pankhurst Institute, this beautiful, quite new building. So the Pankhurst Institute is part of the university, but very interesting. The focus, as you can see from this picture, is how do we integrate, how do we network across a series of partners to deliver improvements in digital healthcare. So you can see some of the partners there, and here's the vision. Enhance the health, well-being, and economy of its citizens, essentially using digital and AI and other related technologies. So some specifics, and you can see here where causality is relevant. So things like prediction and personalized diagnosis, bringing safer interventions, which requires a good understanding of causality around those to provide the evidence for those interventions. So lots of things going on in Pankhurst, and there's always lots of opportunities to get involved with the Pankhurst Institute. So that was all I wanted to say as an introduction. So I think, is there any quick questions? Oh, okay, good. So we can now move into a series of talks from different people around the community. First up, we have Eliana.
Hello, everybody. As it's been said, I'm Eliana Vasquez Osorio. I'm a computer scientist working with in radiotherapy for 20 years now, so quite a while. I worked such a long time with physicists and so on that they have kind of adopted me as one of their own. Every time I tell, like, guys, I'm a computer scientist, I will see like, what? But never mind. So I have been very much, I also got very involved and very interested about causal inference. And I'm very, very happy to be part of CHAI with Matt. And I want to tell you a little bit about what we can actually achieve when we actually introduce causal analysis or causal inference in radiotherapy. So, but before we start that, we need to talk about what is radiotherapy. Okay, radiotherapy is a treatment for cancer. And then cancer, we all know, is a disease that can occur everywhere in the body. And what is happening is simply that cells start reproducing like crazy, right? So there's some damage in a cell, they become abnormal, they start building up, reproducing, and they kind of stop listening to the body's normal signals to say, okay, you are damaged, you need to disappear, right? So that's kind of what cancer is. And if you go to NHS, it says 1 in 2 people will develop some form of cancer during their lifetime. So it's not something that is happening to someone far away. I think we all know people who have been affected by cancer, or maybe ourselves, right? So it is something that is touching us in every single time in normal life. Cancer can be treated with many different treatments, including surgery, chemotherapy, and radiotherapy. And I am part of the radiotherapy side that is using radiation to try and kill that. Before that, just to give a full context, these are numbers from the CRUK. Per year, for every year, we have more than 385 years. So that means actually more than 1,000 cases per day. If you see the number of between cases and deaths, it's not like a big differential there. So it's kind of 50% will not survive, although we're doing much better. So survival has really doubled in the last 15 years, at least in the UK, which is great. But there's a lot of things going on, and I think we can, from our technical side, we can still help a lot to impact this. So what is radiotherapy? As I mentioned, is treating with radiation. So it's ionizing radiation that you aim to the places where there is cancer or where there are these abnormal cells. And what radiation does, it comes and damages the DNA. So like in this bit, so damages the DNA. And then the cancer cells, we talk about them being like these abnormal cells that are just trying to reproduce. And then like, you are damaged. Yes, I don't care, I'm going. Reproduce. So they cannot repair themselves so good. And there's a moment in which they are so damaged that either they die naturally or the body will come and then eliminate them, right? So on the other hand, you have radiation also damages healthy cells, right? So the cells that are healthy will be damaged, but because they are kind of a normal state, they realize there's a damage and then they can either repair it or they say, you know what, I'm too damaged, I'll stop here, and then they are eliminated. So if you put too much dose in the healthy tissue, so in the healthy cells and therefore tissue and therefore in the organs, then they stop working. And that's kind of the counterbalance. You want to kill as many cancer cells as you want without giving too many side effects to them. So to be able to do this, to be able to give radiation to the cell, we need to know where the tumor is within the patient. And that is actually the first step we do. So when we are talking only about the planning of the radiotherapy. So what we do is we take a medical image of the patient, and here you can see a CT scan, so a computer tomography image. And what you see in bright is bone, and what you see in kind of gray is fat, soft tissue, and everything else. And in black, you see the air. What we're looking here is a picture of a patient, of a head and neck patient, a patient with head and neck cancer, sorry. And what you have is a cut like this, right? So we are seeing this is the cranial, this is the brain, this is the part here of the breathing, respiratory waist, the mouth. This person has some amalgam, so some teeth things, and you see the vertebrae and so on. So now we have a full image, which is actually 3D. So you could, if you could go, we could actually see the whole internal anatomy of the patient, and then like that, we can finally find where the tumor is. So that is the first, after you get the image, you identify where the tumor is, the cells, the macroscopic tumor, so what we can see in the images. In this case, is Rob has entered the waiting room. Perfect. And my connection is unstable. So the brain pituitary, the spinal cord, muscles, orthoparity, all these beats are beats that we know we need to protect. We need to try and spare, right? We need to try and keep those as low as possible to avoid side effects. And then the next beat is to plan the treatment, right? So again, we want to put as much dose or a lot of dose in the tumor to kill all the cells and keeping it. So before we see how we plan the treatment, let's see how it is done. This is a very old picture, guys, so I'm sorry for that. They look a lot more fancy right now. But at the end of the day, the same thing. So this is a LINAC, a linear accelerator, that is going to create the radiation, and it's going to be aimed at the patient. So it comes, and then the thing can rotate around, right? And what we actually do is we rotate the image around, we rotate the machine around to build up the dose. So if we see the red contours here are where the cancer is, and then what we are doing is kind of building up the dose, giving a bit of dose from the different directions to be able to put a lot of dose inside here and very few doses in the organs at risk. You see? So that's how it works. And because we have a very beautiful mathematical representation of the patient, so there's an image with the contours, and we know how radiation works because we have the CT scan, so we can actually very nicely simulate and therefore very nicely optimize from where the radiation should come, right? So what we want to say is we want to give the radiation to the tumor while sparing the organs at risk. So we have a 3D dose distribution. Again, this image is 2D here just for seeing it. But in reality, if you were to walk around on top of the patient, you could see which beats are getting the most dose and which ones are getting less dose, right? And that is how radiotherapy is being planned. So it's always a balance between giving a lot of dose to the tumor but keeping the organs at risk in a safe enough threshold that we know this patient is not going to develop bad side effects. And I say bad because that's it. And this is a very cool thing that we have in radiotherapy. We have the power, we have the potential to beautifully fine-tune where the dose is going. It's incredibly precise. And this is an example, I'm sorry for the resolution, from a treatment on which there is, again, head and neck. Sorry for the picture, but we are cutting now in this direction, the axial image. And you see the tumor is around here and there, and there's some bit on the top. And we have identified this little region down here. You see it here. It is a constrictor muscle, so it's related to swallowing. And then you see how it is before, and you see how much we can actually protect that little bit of the organ, right? So if we know that this little bit of the anatomy is key to be able to have good quality of life after the treatment, that the patient is not going to develop problem swallowing, problem speaking, hearing, and so on, then we can very nicely fine-tune treatments. Not always, not for every single patient, but in general, can beautifully fine-tune this dose distribution, this radiation going around. So that's a little bit. So again, how can causal analysis help us? Well, the first one and the most straightforward is, if I give dose here, will that cause that side effect, right? It's a very simple one. If we know that, then we can actually spare the little bits that we want. if we give those here, well, that means that my patient is going to live longer or shorter. This kind of to decide safe thresholds to be able to do treatments. We can find the best ways to plan the treatments for the patients. So normally the patients, as we were seeing before with the Joe, I think was called, the heavy smoker and hypertense and so on, that's how much the patients will come into the clinic, right? They are not this healthy guy, super fit, hey, I got a concept, get me ready. Normally it's older people with blood pressure, diabetes, and so many things together. So then when you have all those things together, you want to be able to find the best way to find a treatment. And I say this because I think it is essential. There are beautiful trials going around. There are many, many more trials that have never, for example, finished recruitment. They're kind of futile, if you could call them. And there's all this beautiful real world data because we have images of the patients, we have blood tests, we have information about how they are feeling, how they are going, we have follow-up, we have all this beautiful observational data. It's a data-rich field. And it would be great to be able to combine and then to extract evidence and maybe even guide future trials. So in the next 5 minutes, that I think is all I have, I want to showcase 3 different projects that we are running in the group right now. So the first one is those very small part of the brain that is connected to some kind of interesting outcome. Second one is how we can actually use causal inference to revisit published analysis to try and estimate, to really get the most out of that, and then estimate the effect of treatment combination, in particular chemotherapy. So I've been telling you about head and neck because that's pretty much where I work a lot, right? So head and neck, when you have treatment, you will irradiate the head and neck region. And then often the patients can develop something called dysphagia. Dysphagia is problem swallowing, right? That simply means like people will have trouble eating, swallowing, talking, all the stuff. And the problem with swallowing is that it's a rather complex thing that we actually do. So if you take the water, they go inside, these guys close, they start going down, this little bit has to move to close this to let the water go down. So it's a lot of muscles moving at the same time. There's a timing involved and so on. And if they move wrongly, then you can have that water is coming into your windpipe, and then you choke. That's the word. So you choke, right? So that is when you have problem swallowing. Patients with head and neck radiotherapy, most of them will develop some type or some degree of swallowing problems. And if it reaches the 1 year after treatment, that problem is going to stay for the rest of their lives, right? And this impacts quality of life heavily. People will stop going to eat in a restaurant, they will stop going to eat in other people's house. They will not be okay when even just taking some watering from other people because they feel completely bad, right? This is all the psychological and emotional part of it. And they can also, because of that, they can lose weight, they can lose nutrition is heavily impacting the life of the survivors. And then if I say, guys, you have problem swallowing, then the first thing you can think about is, okay, the problem is everything that is connected to the swallowing, right? So all these beats that move around, so that is like the constrictor muscles, the larynx, the tongue, oral cavity, the mucosa, blah, 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 blah. And there are actually many nice trials that have gone and demonstrated that if you spare the constrictor muscles, then patients have less trouble swallowing, right? We ran some analysis in our group, so looking at the spatial kind of spatial distributions. And then we found out that actually this little bit here, this is the brain, remember, so this is the brain stem. This little bit here was coming as highly associated with dysphagia. I was running this work with some head and neck consultant. The first time I showed this to the head and neck consultant, he looked at me and he said, Eliana, I think you need to go back and check your stats because that doesn't make any sense, right? It was to the point of I did that. I went back, I checked everything out, and then the results just simply came back being the same. That region down there is associated with swallowing problems. He went and he talked to a speech therapist, they are friends, and then she said, oh yeah, no, of course, we know. We have patients that if you have a stroke in that little piece of the brain, they have problem swallowing, because that is where the swallowing centers are located. So he came back to me and said, well, show me again that results. And then we started looking into this, right? So that was the part. And we came to this via association. It was a very unexpected location. So that's why we thought, okay, we need to start looking at that a bit more carefully. And that is where causality actually comes. We had a talk, this is in 2020, at the end of 2022. So this is the speech therapist. I'm talking, Alan, with short hair. This is Matt, and this is myself, and there were several people online. So there were some clinicians and so on. We came out with a DAC, and then we found an instrumental variable, and we also did backdoor checking, and we were able to find actually some estimation of the cause, which is great. And then I was showing this, I presented this in Estro, and then of course people come and say, yeah, but what if your experts are wrong? And that is, of course, a possibility. Or what if, no. So we actually have repeated all this exercise with US experts. So the only people who sat in the panel were Matt and me, Matt to help me not make stupid mistakes, and me kind of leading the conversation and then working with Dagiti. And the very funny part is that the DACs are completely different. It's like 3 times the number of nodes. We were talking about the sticky saliva. We're talking about the thickness of whatever muscle. I mean, you cannot imagine. We also had surgeons in there, which was a very cool addition. And I cannot present that right now because that has gone right just now into Estro. But if we look at the estimations, they're pretty much the same, because the important variables that are there, like avoiding the paths, are very similar. So the very cool thing is I see it as I'm not a statistician, but I see this as a very cool tool to be able to see, okay, what are we talking about when we are talking about this? To be able to get the assumptions and to kind of model the reality in a simple way that will guide the statistics. So this is one of them, and I'm very, very excited. And this is work from Miran, a student that we have. With Matt. And one of the things is that radiotherapy is heavily reliant on prediction modeling. That's pretty much what Matt said before. So we have many articles and many RCTs and stuff that are telling us, okay, the region, for example, this is also looking at, we're finding that region around here in the heart is associated with early mortality in lung cancer patients. So we can go back to the papers that have been published, again, looking at only prediction, can create the DATs based on the reality behind that study. We can find the minimal adjustment sets, and then we can say whether the values we are seeing there are causal or if they're likely to be biased. So we have been, so Miriam has done these for a couple of papers, and this is just like a subset of them. So we can look at the paper. This is all from the group. What has been adjusted for, and if you were to create the DAC, actually what did you need to adjust for to be able to find the causal effect? And then we can go back and either say, well, this is likely to be biased, or we can actually reanalyze the data, right? So that's something that Miran is doing right now, and I'm very, very excited about this work. And last, Charlie, that is in that chair, that table there, is also looking at how we can actually create, or what is the effect of combining different treatments. So something I didn't mention is radiotherapy doesn't normally come alone. So it's not only radiotherapy to the patients, but there's also some chemotherapy next to it, and sometimes immunotherapy, and sometimes this, and sometimes that. And it's not only if they have also chemotherapy and radiotherapy, but also how it is being combined. So you can have chemo and then radiotherapy, that is a sequential chemoradiotherapy, that's how that's called. Or you can decide to give it at the same time, radiotherapy and chemo at the same time, it's called concurrent. Depending how you give treatment, there have been some RCTs have demonstrated that concurrent radiotherapy is better, right? So the question is, why don't you do it to everybody? Well, because if you are very, very frail, for a person who is our lung cancer patients, they come with comorbidities, they're old, they are weak. If you are very frail, you will have trouble because it's a very aggressive treatment. So most likely you will start and maybe you don't even finish the treatment, which is actually very bad for the outcome. So then the question is, well, the RCTs are telling us that concurrent is better. We're doing sequential. Would it have been better? Right? So the what if question. So then Charlie did amazing modeling to be able to find the individualized treatment effects for all the data. We are using this type of data that has been published before by a group in Utrecht. And then then it's comparing the individualized treatment effect. We can join them to create the average treatment effect. So what happened there. And then we can compare them to what the RCT is telling us. We can compare to what will be multivariable COGS if you put all together and see what comes out. And then it is highly, highly optimistic. We can compare it to what they found in Utrecht with their own data set. And now we have more than 1,100 patients. That's correct. So we can actually also see what is the effect. Okay, if I look at the same kind of reality, how will that change when I have more patients? Will that actually make me come to a different conclusion? And well, that is actually the case. So at the end, the conclusion is, well, we can actually do causal analysis on this kind of data. We can get better estimates or more credible estimates of the effect of the concurrent versus sequential. I forgot to mention, this red line means this way is better concurrent, that way is better sequential. We can then, up to now, if you look at these numbers, you will say, well, you know what, you need to go for concurrent chemoradiotherapy for every single time, but we need to stop and calm down with, we need to check toxicities. So we have only looked at patients' survival up to now, and we need to also include whether the quality of life is being affected or not. And key point is that these will make the basis for the future to create clinical decision-making. So I'm hoping, sorry if I took a couple of minutes more, causal inference, I really truly think is going to revolutionize the way we study dose and side effects. So we will be able to find what are the beats that we need to spare. But of course, our patients are more than just simply the dose. So we'll be able to include all this extra information that we require to hopefully find the best way to create treatments. We will be able to refine our causal knowledge by revisiting findings. That's more a wish than a hope than a reality, but maybe. And will allow us to challenge assumptions in the field and see whether that is actually still the case. And I think all of these together will help us to make better tools to support decision-making processes. And I think that's it for me today.
Thank you. All right. Yep. Over to you, Jason. Sorry, this is pathetic. All right, I'll try. I normally like to kind of walk around when I talk, but that's not going to be me today. Okay, I wasn't planning to show the slide, but Matt gave a sort of brief kind of mention of randomized controlled trials, and I couldn't resist. So I kind of like to think of this kind of like toy history of causal inference, which is obviously a dramatic simplification, as being sort of, since the 1700s and 1800s, we've known about randomization as a source of like understanding the world, but we can't model it in detail. Like, Newton was thinking about was deriving his laws earlier, but the world is kind of often too complicated to admit kind of mathematical description. And so what we fall back on is randomization as a way of understanding effects that we can't model directly. And so, we had James Lind and John Snow in the 1800s. This was formalized in the early 1900s by Fisher. And it was really the debate around smoking and cancer in the 1950s that kind of highlighted the need for us to get beyond randomization as our only way of understanding the world, right? So, you can, it's both like deeply unethical and also way too kind of time costly to do a randomized controlled trial of smoking and cancer. I could in principle do it, but even if my trial was successful, I would successfully kill half of my population. And it would take me many, many years because these trials, like the effects of smoking on cancer takes a really long time. And so that kind of prompted, the massive amount of work from computer science, statistics, econometrics, that has built up this kind of tool set that allows us to estimate causal effects from observational data. And I kind of think that this tool set is like revolutionized. When Joe Dodd was in this talk, I showed sort of in economics, it's kind of it's like completely changed how economics is done. It has revolutionized many of the sciences. So I think it's kind of worth us asking, what is kind of missing from this tool set? As someone who cares about developing causal inference methods. And to me, I think it's kind of really problematic that we don't have anything to say about unstructured data, right? There is this kind of data modality that exists in the world that is increasingly popular, like machine learning works with unstructured data all the time, images, text, and so forth. But we kind of have very little in a kind of textbook causal inference to say about it. And so during my PhD, I worked on methods that at least let us kind of condition on unstructured data. So this was a method known as DeepIV, which was extending instrumental variables to take advantage of deep neural networks. And that let us condition on high-dimensional observations and in principle unstructured data. But it wasn't really saying anything about the sort of causal actors in the system, the treatments and effects. And so sort of during my postdoc and since then, I've been really trying to sort of get at like, well, what can we talk about? If it's either the effects, treatments are kind of harder to think about. Like, what does it mean to be an unstructured treatment? Although we have work on high-dimensional treatments. And so yesterday I gave a talk here at Manchester on measuring kind of interactions between perturbations. So if I do take two drugs, do they have synergistic or antagonistic effects when my measurement is something I can image? And so we've developed some methods around that. Happy to talk about that at lunch, but I thought I'd do something different today. And so today it's going to be it's actually almost sort of more a machine learning talk, but I like it because it kind of uses causal inference for thinking about how to do this well. Okay, so it's a little bit different. But so this is like, how do we pair up multimodal data? So we get some data from different modalities, and we don't have it paired up. And so that's what we're going to look at. Okay. Sorry, animation that we shouldn't do that. Okay, so multimodal data is sort of super powerful in these popular applications that we've seen. So, here I asked ChatGPT last night when I was making these slides for a picture of a man in a blue shirt giving a talk about causal inference at the University of Manchester. And like the image it generates is pretty amazing. But the kind of point is not about generation here. It's more about the fact that it's sort of nailing the concepts, and it's nailing the concepts in relating kind of, there's sort of image level concepts and there's text level concepts, and we have a representation that allows us to connect these two things together. Okay, and how this is done. So how this is done in is you have a representation of the text, and then you use that to generate the images. And this is not a lecture on that, but we could talk about that if you're interested. But the training to get the kind of representations of text and images, the objective itself is pairing up data. So you have a sort of set of images and their associated captions. So here in my examples, it's my man in a blue shirt, picture of tourists visiting Manchester and children visiting the Manchester Museum. You scramble up the images, and you say, find me a representation such that kind of the representation of the text is most similar to the representation of the image of its correct pair, right? So sort of like pairing these data sources up across modalities is the supervision sense signal in some sense. So you sort of find me some representation of each of these respective data. This is the picture from Radford et al. who proposed CLIP, which is probably the most popular of these techniques, although there's a lot in the space. And that's just, it's kind of saying the same thing as I just said in different words. Okay, there we go. So, we have a representation for each of the text captions, a representation for each of the images. Just think of this as some feature space. And we want their inner products to be large if they're correct, so sort of on the diagonal, and small otherwise, right? But this really leverages that we have this pairing between captions and images. And in biological data, which is what we've thought about this most, that's frequently not the case. So what you'll typically do is you'll do some experiments, so you do a gene knockout, and you observe a set of images. So microscopy images, this is something that have another job at Recursion, so that was joint appointment, and they collect these microscopy images at scale. So you can kind of collect a lot of images of cells or a lot of expression data of cells, but you kind of don't typically collect them together. You can collect them separately, but under the same intervention, right? So we've got sort of samples from the same interventional distribution, but they're collected separately because in both cases, actually, often you'll kill the cell in process of imaging it or in process of getting the expression. Okay. Those who know biology well know there are exceptions to this. There are specialized assays that do things well, but I'm just going to sweep that under the rug for the purposes. Okay, so it's a little bit like what we know. It's like we have a bunch of images from Manchester and a bunch of text about Manchester, and now we're trying to solve this representation learning problem, right? And so you could do the same thing. You could do this sort of clip-like thing where you sort of pair up an arbitrary image from Manchester, an arbitrary text from Manchester, and sort of try and pair these up, but you don't know the sort of individual level pairing, and you're going to lose a lot of the shared information between these two different modalities, right? So that's the problem we're going to try and solve. And in order to think about it, we have to get a little bit more formal about how we think about the data generating process. And this is where I sort of start to put my causal hat on. And so the way I like to think about this data generating process is we do have some treatment that we do to the data, right? So in this case, I'm illustrating a gene knockout, which affects some latent cell state, right? So there is a sort of state of the cell that is affected. And then we measure that cell state with a variety of different ways, right? So we might get transcriptomics, or we might get microscopy images, or any of the other, proteomics. There's sort of various ways you can measure a cell. Now, a technical assumption, which I think is wrong, but we kind of makes the math convenient, is we assume that this measurement process is injective. We can talk over lunch over how we can drop that. I would like to drop it, but it makes things it makes it sort of math work out nicely. So we're making this sort of strong technical assumption that we're not losing any information in that process. And then we're also just assuming that each modality has its kind of modality specific randomness, right? So for example, most kind of obviously in microscopy, the specific layout of the cell on the plate is going to be different, and is not going to be captured in the transcriptomics data, right? So there's sort of modality specific information that's not going to be captured in each. And so really, the injectivity assumption isn't that strong if you take that into account. The strong part is that I'm assuming that the knockout is not affecting anything modality specific, right? So I'm saying like when I knock out the gene, it only affects the cell state and not this modality specific information. Okay. Now, the way I like to think about this is sort of by analogy to counterfactuals in causal inference. So we sort of imagine that you sort of can kind of imagine a world where there's sort of these counterfactual views of the data, right? So the sort of cell state is the same, and I could have in principle measured this individual cell using either of these different modalities, but I just happen to choose microscopy, or I happen to choose transcriptomics. And so this is very analogous to what we do in when thinking about sort of first introductions to causal inference. We have some outcome variable of interest. Am I sick or not? And I have a population of people who have either been supplied with the treatment or not. And I could have measured, sort of in principle, each individual person could have gotten either treatment, but I only get to see one, right? So this is sometimes called the sort of fundamental problem of causal inference, is that directly observing the causal effects is impossible. Here's the sort of individual effects, the individual treatment effects, right? Which is this is a terminology thing, which I was going to say. If you get like really sticky about this, individual treatment effects are defined as the difference between the treated and the control, and then sort of you can estimate that, and it becomes a conditional so it's a conditional treatment effect, conditional average treatment effect. We talk about this sticky terminology. People get very grumpy about this, but anyway, so you have this sort of you don't have the kind of factuals in reality to estimate the sort of what would the treatment have done to me, you know, if I had seen this sort of kind of factual, and so by analogy. Here we have, you know, within each treatment group, so these are the different gene knockouts, we have either of the different modalities, sort of, you know, said jokingly, somewhat tongue-in-cheek, you know, fundamental problem of unpaired representation learning is it's directly observing counterfactual pairs or at least requires, I was going to say impossible, it requires specialized assets, right? Okay, so we want some way of pairing up these samples. And the technical challenge is we need to define, in order to pair these up, we need to define a distance metric between these two different samples. But these two things live in very different spaces. So it's not at all obvious, like, how do we even pair these up, right? So we've got like pixel space and gene expression space. You know, what is the right distance between these? It's not obvious at all. And so in order to do that, here's a sort of like brief reminder on propensity scores. So this is from, you know, Rubin in the early 80s. He defines this concept of a balancing score, right? So this is any function b of z. So z is in my notation, it's like, well, these are just arbitrary random variables, but in my notation, this is the latent state of the cell. So some function b, such that when I condition on b of z, z is rendered independent of the treatment. Okay. So like a trivial example of this is just the identity function, right? So if I tell you z, then knowing t doesn't tell me anything more about z. What Ruben showed, if I started to respond, was the sort of coarsest function, the function that kind of throws away as much information as possible, but still keeps this property, is what's known as a propensity score. So this is just the function, this is just the probability of the treatment given z. Okay, so and it's a cute short proof to show that. Okay, so now what we can do is we can say, well, let's take, you know, I said we have this sort of function that maps z to my observation. Okay, so there's just sort of this is some like arbitrary function that captures the measurement process itself, right? So we call that g, it's just a function of z and then the modality specific information, and it spits out this new random variable xi, which is my modality itself, right? And so, you know, that's how we get that. And I can do that on both sides of these terms. Now, under injectivity, I haven't lost any information about the probability of the treatment given z. All I've done is I've just transformed this random variable. And so my propensity is going to stay the same, right? I'm not learning anything new. The modality-specific information u doesn't tell me anything about the treatment under my assumptions. And so all I've done is I've just transformed this random variable. So the amount of information I have about the random variable, if I have this injective map, hasn't changed. And so I just have this, you know, the propensity score is just like a different function, basically, just transformation of z. So I can define it on x instead of z, and nothing changes with respect to the probability of t. Okay, so that's nice. So now I have like two, you know, on either different modality, I can define this propensity score. And these things should be the same if the maps are injective. In practice, they're not going to be the same because the maps aren't injective, but we'll gloss over that. It just gives us a common space to match. So now we've got this, you know, sort of the output of this propensity score defines a common space to match. And it has this nice property that any remaining variation in x is independent of the treatment, right? So if you do a bunch of different biological knockouts, so you do a bunch of different, so instead of just doing a single treatment, that would be a bad thing. But if you're doing like a bunch of different gene knockouts, you knock out all the genes in the genome, for example, then you've kind of
move this, so now it's not just a binary propensity score, it's a multivariate propensity score. And you've moved the latent states around a lot, and anything that's left over is independent of the treatment. And we think of this as being probably not biological information that you want to match on anyway, so we're okay. Okay, so what does the algorithm look like? You just train a classifier on each of the respective modalities to classify which perturbation was applied, right? So sort of take, have a sort of arbitrary, this is like a standard deep learning, take an image of the cell, try and predict which perturbation was applied to it. And just this, you can use your favorite deep learning architecture. And then you apply, once you've trained that, you can apply that and you get sort of a predicted probability for each of the treatments. You do the same thing on the expression side, and you get out a predicted probability for each of the expressions, which treatment is applied. And then you match these things up. So you find the sort of most similar, in this probability space, you find the most similar treated and controlled thing. Right. In practice, we use optimal transport space matching. It tends to be more stable. I could go into the details if you're interested, but entry regularized OT is sort of a nice way of doing this matching and doing kind of a soft matching. I'm kind of going to gloss over this, but there is some subtlety. If you actually want to then try and predict one modality from the other, you have to be careful about how you compute gradients. We can go into these. Okay, so let's just as a sort of example of this. Here's where I said, you need specialized assays. They do exist, specialized assays, if you want to have the paired samples. They just only apply in certain cases. We kind of need this for evaluating the methods, right? So you need to know, it's helpful to know sort of real data where you have ground truth to evaluate the methods, because in practice you wouldn't have this. There's a site-seq assay which will capture surface proteins on the cell and their associated the expression in the cell. So you have sort of expression and proteomics data, and that are paired out. So at a cellular level the sort of relationship between these two, and then you can do these sort of various metrics and see whether which these approaches work best. I don't really want to dive into all of these approaches, other than the sort of the obvious things you might think of doing are like train an autoencoder on both modalities and then kind of match these up in that space. And that works. Those we sort of significantly outperform those. And actually, kind of most interestingly, this approach over here actually has. They do that, and then they have a classifier on top that is kind of. They treat as a sort of regularizer to push together similar cells. They have a kind of hand-wavy explanation, and they treat that as a thing. That's in fact what we're arguing you should be doing. And so we saying like, if you throw away the reconstruction objective and just use that, what was your regularization term, you actually do better on this term. So that was kind of neat. You can do cross-modality prediction, so you can sort of say use samples from one to predict the other. Here we actually on the data sets that we looked at, we actually kind of match the performance, and in fact, sort of it's within confidence intervals, but in fact, like the sort of mean number is smaller, so it's sort of within the range of knowing the ground truth match. And then we did this actually on the imaging data where you don't know ground truth, but at least we can show that our kind of predicted distribution of cells more closely matches the true distribution of cells across modalities. The other last thing that's kind of fun on this is how am I doing on time? I'm tight, so I'll just skip over this. Okay, one assumption that I would really like to be able to relax is dropping this assumption that the perturbation doesn't affect the modality-specific information. That's kind of the thing that's strongest that we're leveraging. It's kind of hard to drop because of known problems from optimal transport. Essentially, you need something. If you want to drop that, you need something like monotonicity. So in 1D, this would be like saying, when I do this matching, you can affect the modality-specific stuff, but only in a way that is kind of monotone. So when I'm matching these up, I'm still going to match the sort of largest to the largest and the smallest to the smallest. The high dimensional version exists. There's a definition called cyclic monotonicity, but I have very little kind of like data generating process assumptions on like when would this arise, and so trying to understand that is worth doing. And finally, just like this is kind of analogous to a matching. This is very much analogous to a matching method, although they're doing very different things to propensity score matching. So this is not propensity score matching. Talk a bunch on what the differences are, but it's doing something analogous in a sense. I'm interested. There was a popular set of work around these sort of cycle GAN methods, which like could do these like crazy zebras to horses translations and so forth with unmatched pairs of samples. If you do this sort of something analogous in this multimodal setting, I think of that as kind of like the synthetic control version of this, right? So for those who come across synthetic control, and it's popular in economics as well, it's like you predict the counterfactual and then you measure the difference between these two things. And so rather than trying to find your best match in your data of the counterfactual, you try and make a prediction of the counterfactual and then measure the difference. So it's interesting to think about like whether that buys you anything. But I think more generally, I think what's fun about this is just it's a nice demonstration of how like causal thinking can lead to algorithms that actually are practical, and it's especially useful when you have to deal with latent variables. So that's where I'll stop. Except to just put a picture of the fantastic interns who worked on this. This was primarily led by Johnny, who was an intern with me last year, and David and Jana helped with getting the multimodal images and cells work going in practice. So yeah, that's it. Thank you.
So once this is once we've got this screen working, so we've got two more talks. First up is Olatunji Johnson from the maths department. And then after this, we'll have Hui Guo talking from the biostatistics department. Then we'll have some brief talk from PhD students. And then hopefully, as long as we open out some time, we can have a brief have a breakout session before going to lunch.
Great. Good morning, everyone. Yeah, good to see you. Yeah, so my name is Olatunji, and I'm a lecturer in statistics in the Department of Maths. So today, I do a lot of work around spatial statistics, so that's why this title is about causal mediation analysis with spatio-temporal data. So today, I'll be talking to us. Probably this is one of the shortest talk I've been in a very long time. And so basically, I'm not going to be going to the maths, but just to paint the idea of the problem to us to see what maybe some people might be interested in the problem we're trying to solve. So mediation analysis has been a topic that has been around for a very long time. And what it means is that you have an exposure, you have an outcome, and you say, okay, there's a variable that actually affects your outcome indirectly, basically. So, and the way this has been done is basically, when this was suggested, when this was proposed, there's really nothing about causal analysis there. Basically, mediation analysis, you want to estimate indirect effect. And you can basically just fit your regression model, and then you estimate this effect. And and this is how it's been done. You have two models, and then you estimate the coefficients, and you interpret those ones, the regression coefficients, direct and indirect effect. As I've said, no causal interpretation. The problem with this, one of the problems was that when you have data that are nonlinear, or nonlinear regression, you can't actually do this kind of things because you can't actually interpret the indirect and indirect effect very easily. Of course, you can do that, but it's very complicated. It's not as easy as just picking the coefficients from a nonlinear regression. So after some time, probably in the 1990s, what happened is that people started thinking about the idea of, if you're talking about mediation effects and things like that, what about causal mediation effects? So that's when the idea of causal inference, what we know as statistician before is just association. We talk about effect, which is fine. But now when we also start thinking of causal effect. So now the idea of causal mediation analysis started thinking. And in the work of Robin and MPA, they actually work out how to come up with this estimate of direct and indirect effects using potential outcome method. And in another work in 2010, actually now discuss the necessary conditions that must satisfy for you to actually place causal interpretation to those estimates that you have. Which, just to refresh our mind about this potential outcome, basically, what the argument was that, let's say you have your outcome, which is a function of your treatment, and then you said, okay, the natural direct effect, you can have it as that equation that I have there. Natural indirect effect as well, you can have the total effect. The good thing then is that you only need to estimate two things. You need to estimate the total effect and the direct effect, then you can work out the remaining effects using potential outcome, which is a very nice framework. And these are the assumptions that must be satisfied for you to actually give causal interpretation to those estimates that you have. And all these things are just jargons. The ones you need to know is that unconfoundedness, you call it reliability, some call it stageability, anything. That is no unmeasured confounder between all these links that you have. That is between the outcome and the mediator, between the mediator and the exposure, between the exposure as well and the outcome. That's what this is saying, basically, which is very difficult to justify, but if you can do it, why not? And justify them. You can place causal interpretation to whatever you get out of it. So let's go to how did I get myself into this problem? So there was a welcome trust ID challenge, and then they said, okay, you want to estimate the indirect effect of climate change on Earth. So the economic impact. So basically, they're treating economy as the mediator, and then they said, okay, if we have all these environmental variables that we're familiar with, and then you have your outcome to the Earth outcome. How do you measure it? They want us to estimate the causal effect. Okay, so we narrow the problem down a little bit because that is very broad. So okay, let's work with disease of despair. That is probably alcohol and things like that. So we narrow the problem down a little bit. So in this study, we chose disease of despair specifically among farmers. And then the exposure, we chose droughts, which is this index here, standard specification, is just a function for droughts, basically. And then our mediators have discussed, we chose the gross domestic, the GDP from agriculture. That's what we chose. There are so many, so the dark diagram for this could be complicated when we are drawing this down. There are so many things that you can account for. So many things. However, when we look at the literature, we understand that there are ones you can find, there are ones that you can't actually, there's no data for it. But because we want to pretend that we know everything, I want to justify the assumption of no unmeasured confounder. Then we said, okay, these two, we know them. You can find data for them. Of course, because I'm not giving you the full diagram here, we have the full diagram that has a bunch of other variables that they are unobserved. So what we want to do is to estimate the direct effect of droughts on this sphere. That is, farmer committing suicide, for example, what's the effect of climate change on that? And then we want to also study mediated effects via the economy, because of course you know that it's not really, it might not really be the climate change that's causing farmer to have mental its challenges. It might be as a result of other complications of climate change. So this is simple as that diagram that we'll come up with, not the big one. The big one, because I don't want to discuss that. That would take me probably a day to discuss, because it takes us like 2 weeks to discuss that. So this is the simplest version of it, which is the variables that we have. But as I've said, this is what we are interested in. We are interested in this indirect effect, the blue line. And that's what we're interested in, the indirect effect. That's what we want to really quantify. Now, the problem we have is that we're able to collect data in space and in time. So we have data on these variables in space and then over time. that is where another challenges come from. So how do we deal with that? So I'm not going to tell you what you've done, but I'm going to tell you just bring the problem in our mind. So these are the problem. Think about we have two close regions that are peers. So this bit is one region and this bit is another region. Also, just think about 2 time points, last month, this month. So this is probably last month, this month, last month, this month. So basically, I'm taking 2 time points and I'm taking 2 districts, for example. And you can see how complicated the diagram that can look like. And this is not a full diagram, this is just something that we can easily understand. But there are so many things, so many other complications with spatio-temporal data, because you have data over time, you have data in space. And one of them is spatial interference. That is, the exposure of one district will affect the outcome of another district. The second one is spatial confounder. We know about confounder, but you know confounder could be broken into 2. One, the unmeasured one and the measured one. And then another complication is that it could be spatial if that confounder itself has a spatial dependence in it. So that's what we call spatial confounder. Of course, probably if there's spatial statistician here, if you hear spatial confounder, you might be thinking of another thing, which is when the predictor and the random effect are correlated. That's not what we are talking about here. We're talking about when the confounder itself, either measured or unmeasured, has some spatial structure in it. And of course, the last bit is the spatial dependence. That is, even this data, for example, if you think about climate data, for example, they are correlated in space. They have their invariant correlation. And how do we adjust for it? And you can look at this line. Those are the black line that I draw. That is, in space, things are correlated. All these variables are correlated. And even in time, they are correlated. So how do we account for all of this in a model? So basically, just to summarize, those are the problems that we encounter and more other problems. So just to summarize the problem, so spatio-temporal data, if you're inferring causal inference from spatio-temporal, poses so many challenges. One of it is spatial interference, which I've discussed, partial confounding, the invariant spatial structure in the data as well, the carryover effect over time. You need to account for that. You can see the data could be actually complicated. And justifying this assumption is a pain. You can't, it's difficult to actually justify like the assumption of unmeasured confounder because there's so many things that you don't know. So that's the end of my talk. Thank you very much.
The last longer talk, which will be given by Hui on causal networks using genetic instruments.
Thank you. Not that long, just 10 minutes, I think. So I'll try my best. So the aim is to understand the complex relationships between exposure risk factors and their causal effects on disease risk. And then this is to ultimately gain insight into causal mechanisms and to develop targeted treatments. So the challenges I or my group encounter often dealing with high-dimensional data, especially biology, omics types of data. So that's why computation is really expensive, sometimes prohibitive. And causal inference and machine learning are now often considered independently, in my opinion. So yeah, people doing causal inference without thinking about machine learning and vice versa. And so in particular, so you have noticed in machine learning areas, people more focused on prediction rather than drawing causal inference. So that's why in my talk, I will introduce how we might be able to combine the two together. This is the triangle. So we are interested in the red arrow, the causal effect of exposure on the outcome, but often from observational data, we have lots of confounders that need to be considered. So we do have this kind of statistical techniques, for example, regression modeling and also propensity score matching, for example, but we cannot actually deal with unobserved confounding without further thinking. This is Mendelian randomization. So yes, a couple of people already mentioned these techniques, but just in essence, it mimics randomized controlled trials because our genotypes are inherited from our parents by random. So using that sort of idea, we're trying to see whether this randomization by nature could be potentially used as a proxy of randomization by investigators to draw causal inference. And then the idea is quite simple. So we've seen the triangle structure. The only thing I added now is the genetic variance, which is associated with the exposure known. So you would not see this genetic variance being directly associated with anything else in this diagram. But obviously, we're saying by mimicking randomized controlled trials for sure, the stronger the association between genetic variance and exposure, the better. So what it does is normally it requires two sets of association analysis. The first one is between the outcome and the genetic variance. So you will have to estimate the effect of this genetic variance. And the second association is between the exposure and the genetic variance, and then you do the ratio. So that the math is quite simple. But what about, as I say, complex disease mechanisms? We have multiple exposures, multiple causal pathways, comorbidities, and so on. So yeah, this is a bit funny. So you will not see any, variables, but there are three different layers of four. Let's say four. So the bottom one is the genetic variance, exposure in the middle, and we have a bunch of confounders and the outcome. So this work was done with my former PhD students. So we have some nice work. So using individual level data, because traditional Mendelian randomization always use summary statistics. So we use individual level data because now individual level data could be accessed easily. For example, even for the UK Biobank data. So that's why we did some sort of nice work on Bayesian Mendelian randomization. But this could be only used to deal with small to medium causal networks because Bayesian approach in general is quite computationally expensive. So that's why if we come across with something bigger, we have thousands of variables in consideration. We do not know structured data, and then we cannot actually pre-specify a model. So we cannot draw such DAG upfront. We can't do it. So how do we do that? That's the idea, how we bring the idea of machine learning. So basically, we look at the PC algorithm. That one is quite commonly used in the machine learning literature, trying to identify large causal networks. So basically, you will see we have the ground truth, which is on top left. What it does is to start with fully connected graph and then trying to figure out conditional independence and marginal independence properties, and then in the end trying to learn the structure in terms of the directionality of those associations. So yeah, if you are interested, please have a look at this paper. So that's the nice work being done by colleagues Gleimer and et al. But limitations of the PC algorithm, first, it does assume no unobserved confounding. So that's not something we would be happy to do, because in reality, no matter how many variables being measured, still, we are not really sure if there are unobserved confounding. And second, we tried this algorithm for our gene expression data, it failed, because we have basically 20,000 genes. And also we have lots of millions of genetic variants, so that's why it failed to run for a very large network. So then we considered another approach is called dual PC. So that this technique was developed by colleagues in University of Basel. So what it does is, so what's so nice about this algorithm is that so instead of the PC algorithm, so starting looking at a pair of variables being conditioned on nothing, one variables, and all variables. So dual PC, what it does is to start from two ends and gradually moving towards the center. So that algorithm is much faster than the PC algorithm. This is just the math behind is to try and calculate partial correlation, looking at the precision matrix. So basically, we combine the Mendelian randomization with the GeoPC, and we come up with algorithm called MR-GeoPC. So we use this method to look at causal networks of gene expression and hypertension. Sample size about 450, and we've got 1.7 million genetic variants and 17K genes looking at the p-values. So yeah, that's what we get in the end. And then we did the clustering because we know the genes, some genes are correlated with each other. So we would like to scale our data in a sort of more manageable way. So after multiple QC steps, we have 98 clusters of genes. And for each cluster, we build a causal network. This is one of the examples. So we started 173 genes and about 1,100 genetic variants. So this is the final causal network we have. And you will see we can look at the magnitude of the causal relationships. So this, and the red node is the hypertension. So that's something we're really interested in. But obviously you will see some of the genes might be weakly, on the path, so associated with hypertension on causal pathway, but some of them, they have quite strong signals. So you can see the thickness of different edges. And also some relationships, you will see bidirectional relationship. That's where we cannot really detect the directionality. We consistently see there is association between the two, but we cannot really tell the directionality. So that's why keep the bidirectional edge between the two nodes. So what's so nice thing about is talking about the speed. So in comparison with the existing methods, so our methods actually is on average about 100 times faster than the existing method. So that's something perhaps a key point of it to sell out of this piece of work. To summarize, causal network analysis using genetic variants has the potential to explore disease mechanisms, and the algorithm we developed, MR-GeoPC, is fast in constructing very large causal networks, but still requires data partitioning because we did cluster gene expressions. So that's why still we find it's quite challenging if we combine integrate lots of multi-layer omics data all together. That would be quite challenging. So this is still something we would like to do in the future. And then if you want to have a look, so this is the paper and all the results code are there. And in the last, I would like to acknowledge our funders from British Heart Foundation and also the Children Institute. Thank you.
So we're now on, we've just got some rapid fire talks from a couple of people in the room. This one doesn't have a name on, but it looks to me like Bowen's slide. Bowen's just trying to find his way through the maze of chairs and tables. You have to come to this one.
Oh, I have to come.
Yeah. Otherwise, the microphone, I'm not sure will pick up. Oh, it might do actually. But anyway, that's okay. So just a couple of minutes.
Hi. I'm here to. Actually, I'm not a PhD student, but I'm here because I'm working for MathTech basically. And I'm interested in prediction and invention, like everybody else here. So for this group, this is the NHS Health Check, and it is a national program, and its target population is aged between 40 and 74. I'm not saying everyone here is satisfied this age condition, but it's good to know. So a recent result published in January 2024, they tried to find out whether this health check is useful. So what they did, they used like a case control study. They found in the UK Biobank data around 48,000 of participants, and the result came out. They have a lower all-cause mortality in CVD, and also have a higher detection rate in other long-term conditional disease like type 2 diabetes or kidney disease. So at least one takeaway message, if you do receive a notification from NHS, please do to do so. And so now, like, to look closer to this ground here, there's two parts: risk assessment and lifestyle risk management. I use some term to sort of interpret it. The first risk assessment is some sort of risk prediction. That is actually just based on the QRIS model, QRIS-2 model actually, and the lifestyle risk management, that is sort of like another study. They're doing like randomized control trial to assess the smoking effect or the physical activity. They seem like doing a separate study, and in my understanding, they should really be incorporated as one, because in the prediction model, what we treatment is sort of as it is problematic. We have this term called treatment initiation, which refer to individual who take treatment after during the follow-up. If we do not do something about it, the result can bias, underestimate the true risk. And for the risk assessment part, that from the RCT, that is always limited in the term called transportability concern, that say the valid causal inference is only limited in that recruited sample or population. So what I want to do in the future is to say the term is called, now it's called prediction in intervention, or it's just the causal inferences. That's saying I want to build a model or system to assess the intervention effect to all these sort of multiple long-term condition, including diabetes, cardiovascular disease. So by doing so, that I'm just going to bring more terminology thing. It's called basically called agent-based model or the micro simulation. So just explain it in a couple sentences. That's basically saying we simulate a hypothetical individual, what will happen in the following transition state from health to CVD, from health to type 2 diabetes, and want to estimate the transition probability. Then after estimated or simulation one, hypothetical individual, we do the Monte Carlo simulation. So that is the process to create 10,000 hypothetical individual. In that case, we can actually to mimic this hypothetical population and make it close to the realistic population. So that after this order work being done, we can just assess the intervention effect, say, stop smoking, or what if I lose weight, to answer these what if questions. Yes, and that's all I'm going to say.
Thank you, Bowen.
Yeah, so hello everyone, I'm Joe. I'm a second year PhD student in health econ, so it's a bit of a different background to the data scientists, computer science, everyone else in the room. I am currently trying to look at causal impacts of access to mental health care on labour market outcomes. So there's a particular service in England which is called the NHS Talking Therapist for Anxiety and Depression Service. Some of you might have heard of it, might not have heard of it. It's a really cool service, and it's, in theory, a great idea to support people that could be suffering from common mental health problems at different levels of severity back into work, into the labour force, to stay in there to help reduce sick leave and everything. It's really hard to evaluate, really hard to understand what we're getting out of the money that's getting put into it. Billions of pounds have been invested into it constantly. More investment is going to be coming as well. If you've seen the news recently, we're really interested in this loss of productivity associated with mental health, right? So I'm generally trying to support essentially this massive literature from RCT studies about, investigating individualized treatment. So talking therapists and psychological therapists, for anyone that doesn't know, sort of things like cognitive behavioral therapy and counseling for depression, I think self-help and guided help is in there as well. So this particular service offers not a diagnosis, but essentially anyone that is a clinical case for anxiety, depression, any sort of common mental health problem, they can receive a course of talking therapies that might be in line with receiving medication and things like that. And the objective is trying to get as many people in as possible not trying to diagnose, but get as many people in as possible, treat as many people as possible, try and support somebody back onto their own feet. So they're necessarily using this RCT evidence of individualized treatments and things like that. What we find in these controlled environments, it's really hard to then model when it gets into the real world population, because the way that people respond to mental health treatments and just general mental health, it's completely individualistic, right? So we have massive patient heterogeneity and everything like that. So my overarching research question for lots of different projects is, does extended access and use of psychological therapy support for individuals with common mental health problems increase their economic inactivity or activity? And my major challenge, so when I was asked to do this lightning talk, because you could talk about a challenge, right? So my major challenge of all of these different research projects I have is finding some form of estimator or estimand that makes sense, right? Having a causal effect and something that has a policy impact, because of how difficult it is to evaluate the service. So I now have used observational data and the sort of patient level data set nationally for this service. I kind of sort of think there's two major issues that I have, and that's the technical side and the theoretical side. So from the technical standpoint, when I'm trying to look at what I'm trying to investigate here in terms of general supply and regional supply and individualized treatment, we have these missing values in this data set. So the NHS was really interested in understanding what we can do to support people into work. They want to collect all of this data on economic activity, and then they don't make sure that this is filled in when they're collecting these things. They don't fully complete patient records as they're going through. They might report something one month, and then the next month they have this drop of things. So all of this really important data that it's supposed to capture, that I paid a lot of money for, it's just not there. Right? And that means that we lose a huge amount of this sample, right? So this national sample that we're supposed to be looking at, this real world context that we're supposed to be evaluating, is already minimized immediately when we get access to this data. We can't look at these bigger scales because of that. And attrition. So in RCTs, there is this control environment. And something that really upsets me is everybody completes treatment. Everybody goes through this process for the most part, or they'll discuss about why and the counter and why people don't at the time. In reality, for this service, 50% of patients don't complete a course of treatment. So actually trying to estimate some form of, again, meaningful estimator on this group is really, really tough. And then the theoretical side of it as well, just to quickly finish up, because I know everyone's probably pretty hungry. What is treatment when I'm looking at this? What are we really trying to understand? If I'm looking at sort of an average causal response to a dose of appointments, or individual treatment or anything like that, that's really hard. So the whole point of this type of treatment and mental health problems is the assessor for the service is supposed to talk to someone and say, what do you need? What do I need to treat you with? What's your goals for the treatment? How do I support you? Again, that's massively different for each person. So trying to understand how one element of a treatment might affect all of these different people adds nothing when we try to apply that to expanding a service or expanding a treatment, because you really have to account for this level of patient heterogeneity and selected samples. So our patient level data set is completely selected, it's self-selected into the service. It's selected by the suppliers as well. And that might be that there are lots of people that have need for the treatment but don't want to use it. And there might be lots of people that have need for the treatment and can't get into it and things like that as well. So yeah, lots of problems. Anyone knows any easy solutions, let me know. But thank you for your time.
Thank you, Jeff. Thank you, Gil. So I'm gonna jump straight in and gonna keep this very high level, but today I want to talk about clinical prediction models. So we're talking about prediction here. These are statistical models that take some information about patients and output predictions for clinical outcomes that we think are useful. Like, for example, what risk does a patient have of experiencing a heart attack in 5 years? These models do not perform equally well in all groups. This is by their statistical nature, right? Your model is not correctly specified ever, so it's going to have some bias, and that bias is going to be different in different parts of the distribution. The problem is that as we're going more and more into this machine learning world where models are black box and it's very hard to understand what they do, at least on the ground when they're actually deployed, these things are harder to spot, and it becomes more imperative to try to predict when they're going to happen and do something about it. Especially when those groups are defined by protected attributes, things like ethnicity, gender, age, and things like that, because there's an ethical and legal question there, right? So there's this field that's popped up in the machine learning world called algorithmic fairness that is all about this. Can we identify when these things are happening, and what can we do about it? So it's basically a bunch of machine learning methods. But as usual, in healthcare, things are a little bit different. This is in part because
there's a lot of differences between groups that are considered genuine in healthcare, or at least that we should model. So if, I don't know, women are at a higher risk of experiencing arthrosis than men, then our model should account for that, so that more women get resources and may get less false positives. So everyone benefits. And in that also lies the other sort of key difference, which is that we don't care so much about equality of performance, we just want to improve performance, especially in those underserved groups. So because of that, a lot of the traditional, so this is like a 10-year-old field, so it's not very old, but a lot of the classic algorithmic fairness methods don't really apply to healthcare at all. And we've really jumped for a more data-centered approach into how we think about this. So basically, when you're using some kind of algorithm using data to solve a clinical problem, you should question whether your data is of the right quality. Is there selection bias? Is there more missing data in some groups than others? Is there measurement error? So we think, like, for example, the little device that measures oxygen in blood does not work as well in different skin types. So if we're using it as a predictor, that might be a problem. And also, this is the one I'm focusing on going forward. What if there's other diagnosis? So, for example, diabetes. I think it's a crazy number. It's like, I don't know if it's 30% or 50% of the people that have diabetes in the UK have it undiagnosed. And that's probably going to vary across groups, right? So basically, the focus now is think about these things and think about how they're going to affect your performance or the performance of the model. But what do we do if we know that these issues are in the data? We could always advocate for better data, but realistically, no study is going to be perfect, right? There's always going to be these issues. And what I'm interested in right now is if we have some certainty about what is happening in the data, can we use that as sort of additional assumptions and feed it back to our models so that we can correct for this as best as we can? And I'm really only getting started with this. So this is the third project of my PhD. It's been very convenient that it's only 5 minutes because I can talk in very vague terms. But really my plan for this is to frame it as a causal question, and specifically as a counterfactual. In a world in which diagnosis rates would be equal across different protected attributes, so that's the counterfactual, what would be the predicted risks of the model that we will have then developed on a particular patient? So this relates to causal ideas in algorithmic fairness. There's a thing called counterfactual fairness and path-specific counterfactual fairness, which are very relevant to this. It also ties with mediation analysis. In a way, we want to control for example, ethnicity when we're talking about the diagnosis effect, but we don't want to control it when we're talking about potential genuine effects. I don't mean just biological, but like social. But yeah, as I said, I'm only really starting to think about this, so really happy to talk more about it in the lunch break. Thank you all.